So viruses are notorious for the long history of causing pandemics and epidemics. SARS-CoV-2 is so far the last of a long series of viruses that caused major death toll on humans. And what you're seeing here is a timeline of the major pandemics and epidemics caused by viruses or bacteria that we were able to record in the human history. Time is represented on the right side of the white track here from the 160s up to 2020. And the death toll is represented by the size of these spiky bubbles. Now, if we zoom on just the last 100 years or so, we can see that viruses that caused major pandemics have killed more than 70 million humans since the yellow fever in the late 1800s, and that includes the Spanish flu, HIV, and other viral infections up to COVID-19. But are all viruses really bad? Is life on Earth better without viruses? And what about all the other viruses out there that we don't know of? Maybe we don't know about them because they are harmless, they don't cause us any trouble. Maybe they are beneficial to us. The examples of good viruses from different ecosystems are really numerous. But today I will be talking about these unknown viruses that we study in the ocean and their previously unexpected roles in maintaining life on Earth, from potentially regulating the climate to the early evolution of life. That would include DNA viruses that I have studied during my PhD and RNA viruses that I have studied after my PhD. DNA viruses are mainly infecting bacteria, and their genomes are made of DNA similar to us. RNA viruses, on the other hand, are made of RNA and mainly infect eukaryotes, from simple cells such as fungi and protists to complex organisms such as plants and animals. All the viruses that we studied in the ocean were retrieved from the water samples that were collected during the Tara Ocean's global expeditions that started in 2009 and circumnavigated the Arctic Ocean in 2013. The scientists on the Tara ship systematically sampled the world's ocean at 210 different sites and 3 depth categories. These 3 depth categories were down to 1,000 meters deep. And they also sampled across 11 organismal size fractions, from viruses all the way to small animals. The major goals of this intensive sampling is answering basic questions about our world, such as how diverse are these planktons, including viruses, across the global ocean? And what is the impact of anthropogenic climate change on their diversity, especially in vulnerable systems in the polar systems? These areas of our world are critical for carbon stabilization and loss and are the most rapidly changing part of our planet. This is indicated here by the global temperature anomalies from the 1880s to 2017. In the Sullivan Lab, we investigated more than 32 terabytes of sequencing data from almost 1,000 samples to identify DNA and RNA viruses across the global ocean. And this is also to answer these most basic questions about viruses in the oceans. How diverse are they? Do they play a role in climate change? And what are the evolutionary origins of these viruses? We indeed found a massive diversity of DNA viruses in the ocean, to the extent that our new DNA virus dataset resulted in around 12-fold increase in the number of known viral species or populations in the ocean. So in total, we had around 200,000 viral species from the ocean, from just this one study. These viruses are known to impact carbon and nutrient cycling. And that's because these viruses infect microbes and other plankton in the ocean, and they blow up these cells, releasing their content into the environment. And this carbon, this small carbon, gets taken by other members of the microbial community. So it's just a cycle that keeps the carbon small. However, these viruses can also help with exporting carbon to the deep ocean, where viruses can also stick to the particulate organic matter that is just coming out from the blown cells and make them more sticky. So these particulate organic matter stick to each other, become larger in size, and get exported or shuttled to the deep ocean. And why should we care about this? And why should we care about carbon export? It is estimated that the oceans absorb a quarter of the carbon dioxide that is produced by human. And viruses appear to be central to this biological carbon bomb. And this biological carbon bomb is responsible for exporting carbon to the deep ocean. In fact, in 2016, Guidi and his team published a Nature paper in which they tried to look for what are the organisms that are best predictors of the carbon export to the deep ocean. And to our surprise, actually viruses, DNA viruses, were the best predictors of carbon export at 150 meters, which is basically where the boundary happens for exporting carbon to the deep ocean. So this is shown here on this map by the intensity of color. So all of these dots are basically the sampling stations. And the more red or yellow color, the more carbon export happening in these regions. And the more blue color is less carbon export happening in this region. Among the DNA viruses that were predicted to drive this process, the carbon export, were the phages that infect phytoplanktons, and which are photosynthetic organisms, such as Synechococcus. For RNA viruses, we also found a massive diversity of these viruses in the ocean, to the extent that our new RNA viruses doubled the number of phyla known to date from just 5, so these were the previously established phyla, to 10. So we added these 5 new phyla. And even within the previously established phyla, we can see here all of these brown lines, which represent the new RNA viruses discovered from our study. And all of the gray lines here represent the previously known RNA viruses. Adding our new 5 phyla to the previously established global phylogeny, we get a complete revision of several of the proposed evolutionary origins of RNA viruses. All of these trees and these global trees were built using the primary sequence of the RNA-directed RNA polymerase domains. So the RNA-directed RNA polymerases are these enzymes that are responsible for replicating the RNA virus genomes, and that's what we use to build all of these, or to infer the evolutionary relationship of all of these RNA viruses. However, the 3D structure information of the RNA-directed RNA polymerases can help us resolve some of the evolutionary relationship that cannot be easily resolved using the phylogenetic trees, such as the trees that we have just seen. So what you're looking at now is a 3D structure network, 3D structure similarity network, where our new sequences are colored here in brown, and all of the previously known 3D structures are in different colors here in this network. And what is really exciting about this network is that some of our viruses appear to be the missing link between the enzymes that make RNA using RNA templates and the enzymes that make DNA out of RNA templates. And because RNA-directed RNA polymerase is supposed to be one of the most ancient genes on Earth, and it's thought that it actually existed in a world that is just made of RNA and peptides before there is a need of DNA viruses, before there is the need for any DNA, we are not just tracking the origin of viruses, but we are also tracking the origins of life. So what I hope that I have communicated today is that from potentially regulating the climate to giving us some hints on the early evolution of life, viruses are much more than infectious agents and a source of panic to humans. With that, I'd like to end with thanking everyone involved in the Tara Oceans and Tara Oceans Polar Circle teams. This is a global effort that required a huge team of scientists to obtain samples, collect environmental data, maintain and curate databases, and provide the essential interdisciplinary expertise. Thank you so much.